Hello, I'm Professor Stephen Locarnini from the Victorian Infectious Diseases Reference Laboratory in Melbourne. This morning we're talking about hepatitis B. And the problem of hepatitis B, I just wish to highlight to you the enormity of the global situation with hepatitis B. The World Health Organization, or WHO, has recognised or identified more than 350 million people are chronically infected with this virus. Over 2 billion people on this planet have been infected or are currently infected with this virus. This burden of liver disease results in annually, every year, 1 to 2 million people die directly as a consequence of chronic infection with hepatitis B. Now the problem with hepatitis B is that it's basically a silent disease for the first 10, 15 or 20 years of persistent infection with this virus. And then, unfortunately, the consequences, the clinical sequelae of this persistent infection start to become manifest, mainly in terms of chronic liver disease, cirrhosis, and then, unfortunately, liver cancer or hepatocellular carcinoma. Hepatitis B is a killer. Fortunately, about 10 to 15 years ago, the pharmaceutical industry developed a number of drugs that were active against the hepatitis B virus, and in the short term at least, effectively controlled replication and improved the clinical outcome for our patients. Unfortunately, like with most of the drugs that we use in infectious diseases, the microbes or the viruses of hepatitis B became resistant to these new drugs that were being used in the clinic. And as each new drug was being introduced, we were getting problems of cross-resistance as well. There was clearly a need to develop an educational tool to help physicians and to help our patients be able to respond better to the treatments that were being used and still are being used today. As a consequence of that, a group of us formed CKEP-B. CKEP-B is a computer-based IT platform for looking at the genome of hepatitis B, the genetic sequence of hepatitis B, that allows us to identify whether the virus is sensitive or resistant to the drug that we want to use. It allows the physician to be able to take a sample, extract the genetic information of the virus from that patient, and then CKEP-B analyses that and then gets a diagnostic report back to the physician to be able to guide the physician on the best way to go forward in terms of the next treatment to improve the overall management of the patient. Now, the CKEP-B program was initially started in Melbourne, but we also had collaborators in Europe and in Asia, because hepatitis B virus is a little bit different in each part of the world. And so we developed this clinical collaborative consortium of people who were experiencing similar difficulties with these treatments in hepatitis B. And so we were able to develop a very extensive, very comprehensive network and a very comprehensive data set of different isolates from different parts of the world with different drugs, so that by the end of the 1990s, the collaborative consortium that was working under the banner of CKEP-B, we had covered all of the major resistance mutations for all of the drugs that were being approved at the time, right through until almost now. Now, what we've been able to achieve from this is a complete coverage of the whole drug resistance scene in hepatitis B. And we decided that it would be commercially sensible to be able to protect that very important data set with some intellectual property. And so the Clinical Collaborative Consortium partners and Melbourne Health developed an intellectual property portfolio, very comprehensive, that covered all of the major mutations and all of the major drugs approved for hepatitis B and in the context of drug resistance. Now, this is critical for allowing a physician then to be able to go to the next step in patient management, to be able to say, you've failed this treatment, we now know that the reason for failure is because of this particular mutation. We now have these new drugs, and we now have identified that if you use drug A against this particular mutant, then you will be able to go forward and improve your patient management. Since CKEP-B has been so successful, we've decided to also apply the same sort of technology, the same sort of approaches to hepatitis C. Hepatitis C is also a bloodborne virus that we see in similar prevalence to hepatitis B. Roughly 160 to 180 million people worldwide are chronically infected with hepatitis C, and the epidemic is actually growing. Now, the other exciting initiative and opportunity for us here in the Evivar group is the strategic alliance with a group called ABL. ABL have addressed the issue with HIV drug resistance, and the complicated use of combination therapy that is required to manage HIV. And I think that they've also been able to develop sophisticated computer programs that not only identify the mutations like we do for C-hep-B, but also because of the very complex drug-drug interactions that we see in HIV, they have got very good educational programs to be able to value add the HIV component of the ABL platform. So I see the future as very, very exciting, because medicine is getting more and more complicated, more and more sophisticated, incredibly dependent on technology. Any tool that actually is driven by science and the bioinformatic process of the IT platforms like ABL, like C-hep-B, like C-hep-C, require an interface that allows translation into simple diagnostic, clear messages to the physicians. And this is what we've been striving to do with both C-hep-B and C-hep-C. Thank you.